to Chapter 4, Part 1. So we're going to break this into two parts. I'm going to kind of focus the first half on prokaryotes, the second half on eukaryotes. So the second half will be doing some drawings and talk about some things, a little bit more. So in the first one, we're really just going to focus on bacteria. So let's get started. Just remind ourselves that there are two groups of cells out there. They're the prokaryotes and eukaryotes. Remember that prokaryotes are things like bacteria and archaea. Archaea are things like plants, animals, fungi, and protists. Remember that. So cells, they all contain the four organic molecules. They contain proteins, lipids, nucleic acids, carbohydrates. They all metabolize food. They all can make proteins, and they all can store and use energy. But they are going to be very different in the structure of their cell walls and cell membranes and whether or not they have organelles or not. So when we get into prokaryotes, that's what we're going to start with, is pro meaning before, or and then karyon referencing nucleus. So that means pre-nucleus. Karyon kind of gets its name from kernel, pre-kernel, the nucleus. Okay. So the DNA is not enclosed by a membrane. Instead, there's just a nucleoid region. That is, the nucleoid is the region where the DNA is. There generally will be a single circular chromosome. There are some species with more than one chromosome. They will use binary fission for their reproduction instead of mitosis or meiosis. There will not be histones in A&P in general biology. We learned about histones. Histones are these set of proteins that are found in chromosomes. The DNA wraps around the histones. Histones, the presence of histones is an important thing when it comes to eukaryotes versus prokaryotes. And remember, there's no membranous organelles in a prokaryote. Prokaryotes are not going to have what is referred to as things like a Golgi and endoplasmic reticulum. They're not going to have membrane-bound organelles. There's the bacteria. They have peptidoglycan in their cell walls, and the archaea that have pseudomurin. Okay. Now we're going to start with eukaryotic cells. Eukaryotic cells, they have a true nucleus. That's what the word eukaryote, U means true nucleus. True nucleus. The DNA is housed in the nucleus. There will be histones. That makes a nucleosome. Nucleosome is DNA wrapped around the histones. We have a nucleosome. They will have membranous organelles. For example, the endoplasmic reticulum, the Golgi, and that kind of stuff. If they have cell walls, it would be a polysaccharide cell wall. Plants, they have cellulose, and fungi have chitin in their cell walls. And those are polysaccharides, if present. There will be a mitotic spindle used in reproduction, so mitosis is something that happens. Now, we can look at cells of bacteria, and they take on many morphologies. Morphology is the cell shape. The morphologies can be bacillus, that is a rod-shaped. So, for example, we have Bacillus subtilis. That is an example of the genus Bacillus. Many of the rod-shaped bacteria live in the genus Bacillus. Okay. Coccus, that's spherical and spiral. Could be a spirillum and a spiral. Vibrio is a curved rod, and a spirochete is a flexible spiral. Okay. So cells can be cocci, bacilli, or spirillum. Okay, Vibrio, spirillum, spirochete, for example. Okay. All right. Now cells can take arrangements. Arrangements are the different ways cells, when they divide, when the cells divide, they will stick into different arrangements, the grouping of individual cells. For example, if they are by themselves, they are called a single. When division happens in a plane, it could be diplo or strepto. Diplo, there's two cells, streptos in chains. For example, a diplobacillus or a streptobacillus, a streptococcus. Okay? In two planes, there's a tetrad. Okay? Tetrads are fours, square of 4. Okay? If it's 3 planes, this will make sarcinae. and cocci arranged in cubes of 8 is called sarcinae. If it's random, we call those random planes. That will give us clusters, staphylo, like staphylococcus. Staphylococcus, the staphylo. Staphylo is clusters, like grapes. Okay. So diplo or strepto. Okay. Tetrads, sarcinae, and staphylo. Okay. Singles, diplo, strepto. And then there's a coccobacillus. It's kind of weird, called a coccobacillus, which I didn't get to talk about here. I probably will at some point talk about coccobacillus later on. I was kind of working in my notes. This is kind of my brand new notes. So, bacterial shapes, they can be monomorphic or pleomorphic. Okay, if they're monomorphic, they are single shape. Most bacteria are monomorphic. Environmental conditions can change the shape a little bit. There are bacteria that can take up many more shapes, like Rhizobium and Corneobacterium, called pleomorphic. Okay? Basic bacterial structures we'll talk about that are associated external to the cell wall is the glycocalyx, the flagella, some axial filaments, fimbriae and pilla. Okay? The fimbriae and pillae. We have a special pillae like a sex pilus or conjugation pilus. Fimbriae are sometimes referred to as your smaller pillae as well. So we'll talk about them a little bit. Now we'll start with the glycocalyx. That's the stuff that surrounds it. It's a sugar coat, I like to call it. So it's a sugar coat. So it's sticky, polysaccharides. There can be polypeptides or both in it. The capsule is the substance that's stuck on the cell wall. This can actually prevent phagocytosis of a macrophage, can try to prevent phagocytosis of the capsule. A slime layer is a loosely attached, unorganized substance. And then there's what's called EPS, an extracellular polymeric substance. It attaches cells in a biofilm to the environment and helps protect cells. We talked a little bit about biofilm is necessary. That's what grows like in your toilet, on your shower curtains, in your coffee makers. Those are biofilms. Okay. Flagella. They are these long filamentous appendages. They can propel. They're used for swimming. They can now, because they look like hairs, we use the word trichus. Trichina, tricho, hair-like. Atrichous means it lacks a flagella. Put A in front of it, that means negative. Trichous flagella. Atrichous. Peritrichous, all over the entire cell. They have many flagella all over. They're very hairy. They're peritrichous. If they're polar, they can have at one or both ends. Okay. So it can be monotrichous, a single flagella at one pole, so we call that monotrichous polar. Lophotrichous, there's a tuft that can be polar, so there's a tuft, like if you have Bio2, you've heard of lophotrochozoans. They have a lophophore for feeding or a trochophore larva. Okay, about to sneeze. Excuse me. There's a tuft, and amphitrichous are at both sides. For example, amphitrichous, you'd be polar because you'd have two poles. Okay. All right. Now, the axial filament of a flagella, that's a flagella. There's a filament that is constant diameter, made of flagellin. It's a globular protein. There's a hook that's part of this, the hook. It's wider, made of a different protein. And then their basal body actually anchors it. And it's made up of microtubules. Okay. Alright, now when these cells move, it can rotate the flagella to run or tumble. Taxis is a word we use to move towards or away from something. If something moves towards, we call that a positive taxis. If it moves away, we call it a negative taxis. For example, there could be a positive chemotaxis when a chemical cue brings something towards it, or a positive phototaxis when something moves towards the light. Negative, when you move away, like a negative chemotaxis, negative phototaxis. Photo means it's a light stimulus. Chemo means it's a chemical stimulus. Okay. A run is in one direction. A tumble is when it changes directions periodically. Okay. All right. Axial filaments. There's an internal flagella called an endoflagella. Seen in spirochetes. It's anchored at one end of the cell, and it's just beneath an outer sheath, so it makes the cell move when it rotates. Treponema pallidum is the thing that causes syphilis. There's another treponemal disease, Treponema yours, that causes the pox. Okay. Now I was talking about fimbriae. Pili is another name for the fimbriae. Fimbriae sometimes are referred to as pili. They are for adherence, for things to stick. They're like hair-like, but they're thinner, more straight than a flagella. They can be at poles, or they can be all over the cell. There can be everything from few to several hundreds of them. Just depends on the species, what they have. There's a special pili called a sex pilus, or a conjugation pilus. I always thought that'd be a good name for a band, sex pilus. The conjugation pilus, or sex pilus, is used to transfer DNA from cell to cell. They're much larger and longer than fimbriae. They're usually only be one or two. It's made of a protein called pilin. Pilin for the pili. Okay. There is the cell wall. The cell wall is more complex. In your bacteria, it is complex, chemically complex, with multiple molecules. In eukaryotes, the cell wall is pretty much only made of either cellulose or chitin, and here it's made up of many molecules. Okay, it's responsible. It's in almost all prokaryotes have them. There's some that don't. It's responsible for shape. It prevents lysis, osmotic lysis. It anchors the flagella. It protects the plasma membrane. It also contributes to the ability to cause disease because there's endotoxins, and it can let it stick and cause disease. That's where the secretion systems often are part of the cell wall, or at least attached to the cell wall. This is where antibiotics work, especially your beta-lactams, your antibiotics that are affecting cell wall, especially for your gram-positives. Okay? This is why it's important that we understand the species. It always, when they give you antibiotics, but they don't know what species it is, they have a guess, but they aren't 100% sure that not all antibiotics, so what they might give you is augmentin. Augmentin has got clavulanic acid in it, which is a beta-lactamase inhibitor, and so it can affect gram-negatives as well. Alright, so peptidoglycan is the molecule that's there. Okay? It is a disaccharide. So this peptidoglycan is in the cell walls of bacteria. Has what we call glycosaminoglycan chains interlocked with peptides, so chemically complex. It's a polymer of a disaccharide. There's many of these disaccharides. Two molecules that make it up is NAM and NAG. NAM is N-acetylmuramic acid, and NAG is N-acetylglucosamine. Okay. These mesh-like layer of sugars and amino acids make up our peptidoglycan. Okay? I'm going to make sure everything's okay. Give me 1 second. Sorry, my phone went off. I had to make sure everything's okay. Alright, now, just as a reminder, the gram stain is going to tell me about the peptidoglycan. Gram positives have plenty of peptidoglycan. Okay? That's because this plenty of peptidoglycan is heavily cross-linked. There's no outer membrane, and here there's teichoic acids that help move cations in and out, prevent the wall breakdown, and help hold the wall together. So gram positives are really good and resistant and tough. As I said, they are the paladins of the bacteria. Gram negatives have a thin layer of peptidoglycan and an outer membrane. This helps prevent phagocytosis, so barrier to antibiotics, helps them invade the complement system. They have LPS and porins that are protein channels, but there are no teichoic acids. please know all the differences in gram-negatives and gram-positives. When it comes to anything about gram-positive, gram-negative, I want you to know all those differences, okay? All those differences. So as we can see here, prokaryotes, there's a cell wall in gram-negatives and gram-positives, okay? And if we see here, there is teichoic acids in the positives and no teichoic acids in the negatives. Negatives have porins, okay? They have the lipopolysaccharides and all that, which has your O polysaccharides, your core, and your lipid A, and all that. The LPS, the lipopolysaccharide. Okay, that would be a thing you would learn in general micro. Okay. Now, the gram stain, remember the gram stain is going to deal with the crystal violet iodine crystals in the cell wall. Crystal violet and grams iodine form crystals. Gram positive, the alcohol dehydrates the peptidoglycan, but it is impermeable to it. Okay? So the crystal violet crystals don't leave it. The cells remain purple. Gram negatives, the alcohol dissolves the outer membrane, leaves holes. Peptidoglycan crystals leave. The crystals leave the peptidoglycan, and now it turns red to the safranin or pink and has that. Now, we also have some cell wall variations. We have acid-fast cell walls. They have mycolic acids, waxy lipid bound to peptidoglycan, similar to a gram-positive. It's mycolic acid. There's the mycoplasmas. They lack cell walls. They have sterols in their cell membrane. They're weird. And there's archaea. They have pseudomurin. They don't have peptidoglycan. Now, one of the things I'm going to end at here is about when antibiotics come in, they damage the cell wall, some of them. For example, penicillin. Okay. Now, what happens is lysosomes digest the disaccharides in peptidoglycan. They are, for example, what tears, mucus, and saliva does. Okay, that's why they're good at helping me fight infections. Penicillin can inhibit peptide, we call transglycosylation, transpeptidation, prevents the building of the cell wall. That makes the cell wall weak. Gram-positive bacteria. This makes them want to lyse. Okay, a wallless gram-positive cell is called a protoplast. Okay, cell wall is destroyed. If it's a gram-negative and it's wallless, that's a spheroblast. Both of these now can be lysed or bursted, and a wallless cell that swells into a regular shape, we just call it an L form. These are what happens when antibiotics destroy that cell wall and it weakens and they start to swell and stuff. This is a common thing that happens. This is where we're going to stop in the first lecture, and I'll pick it up in the next one.